Hello, good morning, friends. We are discussing regarding the cytokines and mainly interferons. Different types of cytokines we have studied already. Interferon is one of them. And interferon, as introductory part, we discuss it is an antiviral glycoprotein which is mainly secreted by either leukocytes, lymphocytes, and some fibroblastic cells. So, interferon being antiviral in nature, it inhibits viral multiplication, and whenever there is a stimulation by viral antigen, the cell secrete interferon. So, we are discussing here the different types of interferon as well as induction of interferon and mechanism of interferon action. So we know that interferons are of three types: type 1, type 2 and type 3. Alpha, beta, gamma, different types of, and lambda. These are different types of interferons. So we are now studying here how induction of interferon is there, how the production is there, and which type of the cells produce which type of the interferons. Means alpha interferon, beta interferon, gamma interferon, these are the different types, but which cells are producing these interferons, that we are going to study here. The induction of interferon is in response to viral infection. Whenever the cell get infected by certain viruses, the cells start producing interferon. So here the genes are specific for coding interferon. There are some specific genes which code for this protein. Ultimately interferon is a glycoprotein. So there are some genes which are specifying the production or coding for interferon. These genes do not express normally when there is no any viral infection or stimulation. But when there is a viral infection, the genes get stimulated and expressed to produce interferon. It means that normally, in normal cell, the genes coding for interferon, they are not expressed. Whenever the gene expression is there, then interferon is produced. Means firstly transcription and then translation. Then there is a protein synthesis, we know very well. So, in normal cell, means whenever the cell is not stimulated or whenever there is no any viral infection, in that cell the genes are not expressed, means there is no interferon production. But whenever the cell get infected by certain viral antigens, then these genes get expressed and then interferon production is occurred. There are various inducers for producing interferon. Means certain agents, certain antigens, certain bacterial products, you can say. These are acts as an inducer for interferon production. Means these agents induces the cell to produce interferon. They are called as inducers. Inducers for interferon production. For example, glycoprotein, viral glycoprotein. Viral glycoproteins are responsible for interferon production, means it can act as an inducer. Secondly, bacterial flagella. We know that certain bacteria are motile because they are having flagella, and flagella is made up of protein flagellin. So we know all about the flagella. So whenever there is a bacterial infection, in certain cases, not all, the bacterial flagella can also act as an inducer for producing interferon. Simultaneously, bacterial endotoxins. We know that bacteria are pathogen, viruses are pathogen, but there are certain factors which are helping the pathogenicity or virulence power of the microorganism. Here, endotoxins, certain bacteria are producing endotoxins. So whenever there is a infection to the normal cell by bacteria and if they are producing endotoxins, then these endotoxins can also act as an inducer for interferon production. So it means that not only viruses but also certain bacterial products can also act as inducers. Flagella as well as endotoxin, these are also acting as an inducer for interferon production. Then certain other types of the pathogens, which includes rickettsia and chlamydia. Rickettsia, we know it shows the character between some characters resembling with bacteria, some characters resembling with virus. So certain species of rickettsia, whenever infect the normal cell, that it can induce interferon production. Chlamydia is also one type of the organism or one type of the pathogenic organism. So it can also induce this interferon production. It means that interferon is produced or it is stimulated by certain inducers. For example, viral glycoproteins, bacterial flagella, bacterial endotoxins, chlamydia and rickettsia. These are the agents which are inducing induction or production of the interferon. Now we discuss induction of alpha interferon. We know that there are three types of interferon basically: alpha, beta and gamma. Lambda is also there. So how interferon alpha is produced? How the induction of alpha interferon is there? The production of alpha interferon is dependent on the interferon beta. The interferon beta stimulate the synthesis of immune response factor IRF7, immune response factor. What happens here? The production of alpha interferon depends upon the interferon beta. It is dependent on production of interferon beta. The interferon beta stimulate the synthesis of one factor, which is that immune response factor 7. Whenever the virus infect the cell, the virus infection activate the IRF7, this factor, immune response factor 7. So whenever the virus infect the cell, the IRF7 factor get activated, and which brings about the transcription of alpha interferon genes and production of alpha interferon. So it means that the interferon beta is responsible to some extent, which stimulate the synthesis of IRF7 factor. Whenever the viral infection is there, the viral infection or a virus activate IRF7 factor, due to which there is a transcription of alpha interferon genes and there is a production of alpha interferon. The induction of alpha interferon in virus infected leukocyte is different from fibroblast. So in previous lecture what we discussed, leukocyte, lymphocyte, fibroblast, these are the major type of the cells which are producing interferon. So here this alpha interferon which is induced in virus infected leukocyte, it is different from fibroblast. We know fibroblast is producing beta interferon. So it is different from fibroblastic cell. Leukocyte do not require stimulation by interferon beta for induction of alpha interferon. Whenever the leukocytes are producing alpha interferon, these leukocytes do not require the help of beta interferon. What we discussed just now, the production of alpha interferon is dependent on beta interferon. But here, when the leukocytes are producing, leukocytes are cells, white cells. So these are producing interferon. Their stimulation do not require the help of beta interferon for producing alpha interferon. So they can, on their own, they can produce alpha interferon. They are not dependent on the beta interferon. Then, how the induction of beta interferon takes place? The fibroblastic cells are the major type of the cells producing beta interferon. There are three types: alpha, beta and gamma and lambda. So which cells are producing beta interferon? Fibroblastic cells or fibroblast. So these are the major producers of beta interferon. When fibroblasts get infected with RNA virus, the double-stranded RNA released as a viral genetic material or formed during the process of replication. When this fibroblast get infected by RNA virus, viruses are of different types: double-stranded DNA, single-stranded DNA virus, double-stranded RNA virus, single-stranded RNA virus, like that. So we know that either one of the nucleic acid is there in virus, DNA or RNA. So when the fibroblast get infected by the RNA virus, the double-stranded RNA is released as a result or as a viral genetic material, and it acts as a good inducer for interferon production. Means what? The RNA or RNA virus acts as a good inducer for interferon beta. The viral RNA stimulate translocation of transcription factor NFKB from cytoplasm of infected cell to the nucleus. RNA virus stimulate translocation. Translocation means movement. Movement of what? Movement of transcription factor. Which transcription factor? NFKB. So there is a movement of NFKB, transcription factor, from cytoplasm to the nucleus. And here this NFKB factor promote the transcription of genes. Which genes? Beta interferon genes. So due to translocation of NFKB factor, there is a transcription of beta interferon gene, and when there is a transcription of beta interferon gene, then the synthesis of beta interferon take place. Because we know for the synthesis of any type of the protein, there should be transcription and then translation. So here what happens? Beta interferon is produced by mainly fibroblast. Fibroblast, when infected by RNA virus, double-stranded RNA is released as genetic material, and which is acting as a good inducer for interferon production. Here the viral RNA stimulate the translocation or the movement of transcription factor NFKB. This transcription factor promote the transcription of beta interferon genes, and due to this transcription there is a synthesis of beta interferon. Okay, now understand, fibroblasts get infected by RNA virus. There is synthesis of double-stranded RNA as genetic material, which is going to stimulate the factor. Which factor? NFKB. This NFKB, it is a transcription factor. So this NFKB later on stimulate the production of beta, or it's stimulate in the sense it is responsible for transcription of beta interferon gene. So whenever beta interferon gene get transcribed, there is synthesis of beta interferon. Secondly, the viral double-stranded RNA as well as lipopolysaccharides, tumor necrosis factor and interleukins stimulate kinases for phosphorylation of NFKB also. Means for beta interferon production, it is necessary that there should be translocation of NFKB or there should be phosphorylation of NFKB, so that there is a transcription of beta interferon genes, and then there is a synthesis of beta interferon. Means NFKB, this factor is important here for the stimulation or for the synthesis of beta interferon, because this particular factor is going to stimulate or going to transcribe, you can say, the genes responsible for beta interferon. So lipopolysaccharides as well as RNA as well as tumor necrosis factor and interleukins, they are going to stimulate the kinases for phosphorylation of NFKB, and this NFKB factor promote or it is going to transcribe the beta interferon genes, and then there is a synthesis of beta interferon. So what we discuss here, fibroblast gets stimulated by RNA virus. Double-strand RNA is synthesized as genetic material which is going to stimulate the NF-κB. The translocation of NF-κB factor is there, which is the transcription factor, and NF-κB later on promotes the transcription of beta interferon genes and there is a synthesis of beta interferon. Similarly, double-strand RNA, lipopolysaccharides, interleukins and tumor necrosis factor, they are stimulating the kinases for phosphorylation of NF-κB factor, which is responsible for transcription of beta interferon genes. Now, induction of gamma interferon. How gamma interferon is induced? When the antigen presenting cell present the antigen to Th1 cell or CD4 cell, CD4 means cluster of differentiation, and to CD8 cell. We know that T cells have been divided into two major types, subsets, T helper, T suppressor. So CD4 commonly we call as a helper T cell, CD8 commonly we call as a T suppressor cells. So when the antigen presenting cell means APC present the antigen to CD4 cell and CD8 cell, gamma interferon is produced by these cells. There is a production of gamma interferon by these T cells. The interaction between the antigen presenting cell and lymphocyte means T lymphocytes, leads to signal transduction, and then there is activation of transcription factor to brings about expression of genes for gamma interferon. So how it occurs? The antigen presenting cell present the antigen to CD4 and CD8 and then gamma interferon is produced by these cells. Here the interaction is important. Interaction between the antigen presenting cell and lymphocyte, that leads to signal transduction and thus they are activating the transcription factor firstly to bring about the transcription of gamma interferon genes. So whenever the transcription of gamma interferon genes take place, there is a synthesis of gamma interferon. So this is regarding the induction of alpha, beta and gamma. In next lecture we will discuss the mechanism of interferon action in detail. Thank you.